It's working? Oh, now we're doing it. Okay, today it's a pleasure to have Tamara Terzian, who's a member of the Cancer Cell Biology Program. And just a little background on Tamara. She got her PhD in biomedical engineering from the University of Claude Bernard in Lyon in France, and did her postdoc with Guillermina Lozano at MD Anderson in Houston, Texas. And she came here as an assistant professor in the Department of Dermatology in 2013. Her work focuses on a very important tumor suppressor I'm sure everybody knows about, p53. It was once on the cover of Science magazine as Gene of the Year. So she's going to tell us about some nice studies she's been doing on the role of p53 in skin cancer and melanoma, and about its protective effects, actually, when skin is treated with ultraviolet light. Tamara.
Hello. Thank you, Bob, for a nice introduction. I would like to first start my talk by thanking my lab members for their hard work and making, giving me the opportunity to present the data today, and also my collaborators, especially Nima Sobhani, that he also helped me with some of the data. And special thanks for the funding agencies. Without this, their support, this work would not be presented today, because as we know, research costs lots of money. Also the cores, and especially I would like to thank Bob, because he was my mentor, and especially his mentorship with grants was very valuable. Thank you. So my talk will be divided into two parts. The first part is p53 pathway in development, and the second one is about, the first part is about wild-type p53. The second part is about mutant p53 and its role in tumorigenesis. Just a reminder for the students and that, about p53 pathway, even though this is an audience that would know about it pretty well. p53 is a major tumor suppressor. It is mutated in more than 50% of the tumors. It's activated by multiple stimuli, including DNA damage, oncogenic activation. And for our topic today, ribosomal stress will be of great importance. So once p53 is activated, it will induce inhibition of the cell proliferation through cell cycle arrest, senescence, and differentiation, and also will induce apoptosis. So to keep p53 in check, as you can imagine, you don't want it to be randomly activated because it would be detrimental for normal development and for normal function of an organism. It's kept checked by mainly two inhibitors, MDM2 and MDM4. They belong to the same family. They have similar structure. They maintain together and separately p53 in check at low physiological level that is undetectable normally by immunohistochemistry. And one thing is MDM2 is the main difference between both of them are that MDM2 is an E3 ubiquitin ligase, and it inhibits and degrades p53 and targets it for degradation. However, MDM4 does inhibit but does not degrade p53. So let's start with the talk on p53 in development. So we know that p53 null mice develop normally, mostly, but they are tumor prone. So what happens is, you can see here that p53 null mice can get exencephaly, they can get cleft palate. They're mainly female mice that get this phenotype, and they usually die in utero. But when they are born, they develop normally, but they get tumors around 5 to 6 months old. So if you take off the negative regulators, for example, MDM2 or MDM4, you will see that there will be lethality, embryonic lethality, very early for MDM2, and a little bit later around 7.5 to 9 DPC for MDM4. And what made these regulators as the bona fide regulators or inhibitors of p53 in vivo is the fact that when you take off p53, 2 alleles of p53, when you knock it out, in conjunction with no MDM2 or MDM4, you will have a rescue. So that was not proven much in all the other regulators that you can find of p53, because usually you will not have that rescue. So that's why we believe that MDM2, MDM4 have a primary role of regulating the p53 level, while they do have also p53 independent function. So here you can see that MDM2 null mice with p53 null, or MDM4 null with p53 null, or even the triple, they are alive, but they do get tumors. And all these, they get tumors at the same rate as p53 null mice. So what happens if you have an MDM2 heterozygous or MDM4? Well, these mice are normal. They develop normally. They live like wild-type mice. They do not show detectable levels of p53 endogenously without any stress. However, we can see that if you have these mice and you stress them by IR, for example, here you have the 10 gray and this is the 6 gray, and this is for MDM2 heterozygous or pyromycin, which is a hypomorphic MDM2. You will have activation of p53, which leads to their death. So there is a sensitivity to IR. And the same for MDM4, you will have both of them are increasing sensitivity to IR. And that's due to p53. So if you take off p53, for example, like here and here, you will see that you will have that rescue of the sensitivity. And that's true for also if you have an oncogenic stress like MYC, driven lymphomagenesis, like if you use the EU-MYC model, like here, both in MDM4 and MDM2, you will see that there is a delay in tumorigenesis, and that is due to p53 activation. So there is an endogenous activity of p53 in these models. That's why we now call them the mouse models of p53 function, because they have endogenously higher p53 activity than a wild-type mouse. And when you combine MDM2 and MDM4, during development, you will see the same phenotype like you have no p53, for example, exencephaly and cleft palate. These mice, when they are born, they will be smaller. They will not live in general long time. This is the first mouse that lived until 35 from hundreds of mice, and only one. So they will be usually, they will have that curly tail too, because they have a neural tube defect. And okay. So the other thing is when they develop. So when they are born, they have also postnatal developmental defects. So for example, the cerebellum, it will be rudimentary. The EGL doesn't, proliferate as much, and we have also like lots of death of granulocytes. And the same here for, like you see, this is the wild-type cerebellum, and this is like at P8, 8 days after birth, and the cerebellum is rudimentary, which makes these mice very ataxic. So they will have, some of them, they will have hypoplasia, so they will, of the bone marrow, so they will die from that. And they will have also hyperpigmentation that I'm not showing here. But mainly these were due to high p53 levels, like for example here in the bone marrow, and the caspase-3 cleavage and activation. So you have lots of death in these p53 sensitive tissues. So in summary, we can say high p53 mice display developmental and postnatal aberrations. So we encountered another mouse when we were working. It's a ribosomal model of a ribosomal stressed mouse, which has a lower expression of RPL27A. So it's the normal protein that is expressing, but it's like around 20% less expression, and depending on the tissue. And here are the main tissues that are affected: cerebellum, bone marrow, and skin. And you can see that these mice recapitulate what happens in high p53 mouse models. That means that you have smaller size, you have the bone marrow hypoplasia, you have the cerebellar defect, and you have hyperpigmentation. And as you can see here, MDM2 heterozygous, MDM4 heterozygous, and double head, they will have gradually higher levels of p53, and that's why they're having higher levels of pigmentation, for example. And that's due mainly to high melanocyte numbers, and also like the melanogenesis is increased. So here we can see p53 is staining in two different, you can see one is lower, one is higher, because there's the penetrance is different, and there's different levels depending on the mice. That's why these mice are born, and they live longer, because their level of p53 is variable between mice. And for example, here in the cerebellum you see it, and in the skin too, the green one is highlighting p53. So other models of ribosomal stress also have this p53 dependent phenotype. For example, here the RPL24, and all these red boxes highlight these. But one thing to remember is these are ribosomal proteins, so they also have a function to synthesize proteins. That's why if they are lethal if they are homozygously deleted, for example, or mutated. But at the heterozygous level, they will have smaller size, they have pigmentation dysfunctions, they have also, like some of them, embryonic lethality, and all the pancytopenia, which we see in the different mouse models of p53 function. And so that means that mouse models of ribosomal proteins, which are ribosomal stress, also model p53 function. So in summary, we can say that if you have too little or too much of p53, you end up in the same place. It's not good for your development. And this is a table where you can see that p53 levels is exquisitely regulated. If you have above normal, you have certain defects, and if you have under that normal, you will have certain defects. And the rates, even though it's not shown here, it could be even 5% of difference in p53 level, and you will have, for example, a difference in either survival or between life and death for the embryos. So it is very exquisitely regulated by the two main regulators, MDM2 and MDM4. So we wanted to go further and see if there is genetic interaction. And we crossed MDM2 heterozygous mice that has endogenously higher p53 level with SFA, which is the RPL27A mouse that has lower levels of the ribosomal protein. And that also have p53 endogenously high to push the system a little bit further. And we can see that as p53 goes up, then you have a lethality happening. So these double heterozygous mice, if it's MDM2 or MDM4, they are embryonic lethal. So we could not, as you see here by the zeros, find these mice, and we did many dissections out for the embryos, and we could not find that are past the 16.5 DPC. So what happens? This is a pregnant female. We followed her by MRI, and we followed the embryo's development. And when you have, for example, here, no embryo, that means that that embryo died. And we followed their volume. And you can see this embryo, for example, he started losing weight. That means he's dying. He's already died and losing weight and being resolved by the system at 15.5, and the same here too. So here, what we see when we dissect these embryos, we see that the double heterozygous have vasculature problems, but you can, this patchiness. They are very edemic, like you can see here. Now, the penetrance is nearly 100%, if it's not 100%, but there is a variability in the manifestation of the phenotype. And here there's an example of a frontal where you can see the buildup of fluid. So we tried to grade it. So we did it from mild to severe for edema, and we scored it for hemorrhaging. And we can see that 100% they have it when they have that genotype. And also, like, we didn't find any embryos born beyond 16.5, but mainly 14.5 and 15.5 are where we see this phenotype. And when you take off p53, what happens? You just, these mice develop normally, and they're born. And here is an example of the double heterozygous mice. And they have p53 heterozygous in them. That's why it more normalizes the levels of p53, and these mice are born. So I'm not presenting here today, but their ataxia is not really solved. For this mouse, the MDM2 heterozygous, it is. They are not ataxic anymore, but not for the D2. And as we remember, the D2 here, they are the most severe ones. And actually, one thought came to mind to say today, that this is the first time I see a phenotype that is linked to severity of MDM4 versus MDM2, because the MDM2 is the main, the first actually, and also like the main inhibitor of p53, and they regard MDM4 as like the little brother. So it does do its job, but not as well as MDM2. So normally, if you have conditional knockouts or in the system, nervous system or in the heart, always MDM2 phenotypes are worse than MDM4. This is the first time we encounter a case where actually MDM4's phenotype is much more severe than the MDM2 phenotype. So now we know that it's due to p53 because you take off p53, these are rescued. We wanted to go further because we wanted to convince also our reviewers that this is true. Especially when you go for a grant, you really need to nail it. So we did a pharmacological inhibition using a drug that I thought it won't work, and I believe that the secret in our field that this drug doesn't work. But I still wanted to try, and I gave it a try. I injected the pregnant, actually, Molly, Mima, and I injected the mice daily after 11.5, and these mice were born. Now, these are the first mice that were born. So the first time we thought, okay, there must be, let's repeat it. So we did it for 2, and we have 2 crosses from the MDM2, the MDM4, and 1 cross from the MDM2, and they're all born. And this is the first time we find the birth. Now, they are not perfect. They died after they were born, but they made it with a drug that I wasn't sure that's going to work. So I think with better drug, you probably can rescue a phenotype because you are hitting p53, and the genetic models also support it. You do see the splabbing of the skin. It's loose. There is something called the Noonan syndrome in humans, and they also have the skin, it seems, that is, especially around the neck, that is loose. So we were very excited by this, and here you can see the genotype that show that they're MDM4 or MDM2 heterozygous. So we looked at 53 targets. This is actually a work in progress. We are adding more targets here, and I want to do a targeted 53 panel for by RNA-seq. And we see that Noxa is a little bit higher in these compared to wild type. But also like Puma is higher, but this is actually preliminary data. So we looked at the phenotypes to understand how to analyze this embryo, and there is nothing that we can find in liver, lung, heart, and we had multiple pathologists go through this. So we're now investigating. We have an idea what it is, and we're trying to tease it out, and of course it's p53 dependent. So in summary, p53 activation is detrimental to normal embryonic development, and further, especially activation versus lower levels. And p53 further characterization of the phenotype will help us to delimit the role of p53 in development. Also, we will try to identify targetable p53 targets that for this phenotype, because we can also go on to the clinic, and for similar phenotypes that we can see in human, maybe we can modulate p53 levels just to manage the condition. Because we also have, for example, something called hydrops fetalis, where children or babies are in utero will have something like that, and 50% of them will die. So we are hoping that we can rescue that with just playing on p53 levels.
Before we go on to the next part, because I'm really curious about the fifth one, because like you said, there's a lot of skepticism. I think that you have to rush and have to work.
Yes, I did rush. Because the first time when it didn't rush, it didn't work, because I had to do between. But, oh, okay. Yeah, because it degrades very fast. It degrades so fast, that's why.
Can you really validate that it is, that the effect of the drug, I mean, because the drug can have a real effect, and you still have nothing to do with p53.
Right.
Some other effect, you know, we just don't know about, but you really validate that what you're seeing is p53.
So you want me to validate what's validation? Is that what you're telling me?
Show that the drug is rescuing the phenotype by inhibiting.
Yeah. Well, it's just because, you know, since I have the genetic model that proves it with one allele of p53, I just feel like I don't want to waste money on it too much.
You showed that just like with the heterozygote for p53 targets probably go down, right?
Mm-hmm.
Does the drug also bring down the same targets?
Right. We're collecting these to do that. So we will go there and see what we are doing. But it's, in general, it's a pretty selective drug. The challenge was it was very highly degradable, and you need to use it in certain conditions. So we did really rush for it. Yeah. Yeah, but they have some papers where it depends on the system. I guess in the nervous system, it's working. If you do other system, it's not working. So it was very much tissue specific. That's why I wasn't sure it's going to work, because I actually, in tissue culture, I couldn't make it work. So, yeah, it just like, I was amazed to even have it alive. So I was like, wow, that's amazing. Yeah. Thank you. So the second part is about mutant p53. Now, what I've told you before, it was all wild type p53, which is tumor suppressor activity and all. Mutant p53 is actually the contrary. It's the devil, is the one that actually push for tumorigenesis and for promotion of metastasis. And that's we know from our human data, where you also have more than 50% of the tumors, and some of them even 90% of tumors having p53 alterations, some way or the other. And you will have also like this, the one that I'm going to talk to you about is the 175. It's one of the most commonly used one, because also it was probably the first one that has a gain of function mutation that was described. So you can see here, this is the DNA binding domain. It's where most of the mutation happen. 80% of the mutation happen in this area, and these ones are highlighted here. They are the ones that are hotspot mutations. So we know that mutant p53 is worse than having no p53. So no p53, you have cancer. But mutant p53, you have a gain of function and dominant negative, and you'll have metastasis. So p53 null mouse was not the ideal model to study tumorigenesis for especially if you are looking lymphoma syndrome type of tumors. Because first of all, they get tumors very, very early. And then they mainly develop sarcomas and lymphomas, and they don't have the other type of tumors. And also metastasis is a very rare event. Actually, I never catch one in the many mice I did. But if you have a mutant 53, especially the 172H, and I'm going to abbreviate it to H, it seems to be acting as a better model. That's because not only it represents a missense mutation that's found in tumors, human tumors, but it's also like metastasizes, and I will show you the data. So there was a knock-in mouse, and so we have the homozygous here, if you have mutant 53 homozygous, and you have the heterozygous here. And as you see here, they are the same, they have the same survival and the same tumor spectrum, then you have no 53. But the only thing is when you have heterozygous, you have also a dominant negative in addition to the gain of function. And you also have metastasis, as you can see here. And that we didn't see, or nobody have seen too much in the 53 null mouse. So this was a good model to study tumorigenesis in. So based on the literature, everybody thinks you have a mutant 53, it's stable, it's detectable by immunohistochemistry. That's how you predict the prognosis for what happens to the patient. Where we were surprised to have not detectable 53 in the mouse when it's a normal tissue. So for example, these are two samples of pancreas and bone marrow, and this is the mutant, and we tested multiple, and I'll show you later. And there was no detectable 53 level by immunohistochemistry. However, when you have a tumor, you do have upregulation, and that's something in human tumors you find, but was interesting that you do not have it all the time. You have some tumors that never upregulate at 53, and we don't know why that is happening. And that's an interesting thing to actually investigate. But what's interesting too, that if you have a mutant 53 in the tissue, the metastasis in the lung, for example, will have high 53 level, but it's also not all cells. So something is playing on the levels of 53 in these tissues. And here you can see the data that majority will have positive p53 staining. And so what we take from this is that there are gain of function because there are metastasis, and there is a dominant negative activity that I'm not going to talk to you about to keep things simple. And we have LOH too. So we wanted to know how this mutant 53 is regulated. Is it the same as wild type? Because at this moment, all the studies were done to study the wild type p53, but not the mutant. So we looked at the negative, obviously MDM4, MDM2. We looked at the oncogenic stress, like stressors like MYC and RAS activation. We looked at DNA damage, and we tried to follow how this mutant is activated and what type of readout we will have. And we find that this is published already, that MDM2 null mice with mutant 53 homozygous, they will have, contrary to the one that is normal tissue here, with only mutation of 53, will have high 53 levels. So what is not known is the MDM4 at this moment, and we're trying to publish this. And when you take off MDM4, it's the same. So you will have the same readout, and when you double it, you will have also higher p53 levels. And here is Western blot showing that with and without IR. But interesting, we look at proliferation, and we look at caspase. And we quantified it. And you can see here that if you have higher p53 levels, you will have higher also proliferation. But if you have higher p53 levels, somehow the proliferation goes down when you knock out both inhibitors. Now we have a theory that about this, but it was very unpredictable result for us because we thought it's going to continue to grow, to have more proliferation, but it didn't. And the other thing that surprised us, if you look at the survival, these mice also like seems to be shifting and surviving a little bit, later. And looking at the different regulators, like KRAS, if you have upregulation, oncogenic stress, that's the same too. You will have activation and stabilization of mutant p53. So what does it mean? All this, what does it mean? It's because, what you need to remember that oncogenic stress, if you have a wild type p53, you have no MDM2, MDM4, it pushes for apoptosis and inhibition. That's good for tumorigenesis, for example, to keep things in check. But when you have a mutant p53, you will go to dominant negative, as we saw, and gain of function, because it will go to metastasis. So you need to avoid this, basically. And when you look at, for example, if you try with doxorubicin, which is used in therapy, and you can see that here, this is the mutant p53, and this is the p53 now with doxorubicin. You will have actually earlier onset and death for the patient. So it's very, you have to be very careful considering the therapeutic drug selection. So that's why it is important. It is important to know what we're doing, what's the environment, what type of mutation you have, because also not all p53 mutations are equal. So in summary, MDM4 is a crucial inhibitor for both wild type and mutant p53 in vivo, like MDM2, and increased in mutant p53 stabilization with the absence of both suggests that MDM2 and MDM4 are working together to push p53 down. I didn't show you this, but actually we are thinking there's another E3 ligase inhibitor that can explain why not every cell is positive for p53. And also there are some tissues that do not have p53 accumulation no matter what. Even if you take off the negative inhibitors. So something is compensating in that tissue. So we're trying to do, we did some RNA-seq for the targets of mutant p53 that I'm not showing today, but we also did, we are trying to do mass spectrometry to identify the E3 ligase and see what are the proteins interacting with the mutant p53 that can explain some of these tumorigenesis phenotypes. And thank you. Questions?
Imagine that might be the case for almost any sort of similar, whether it's radiation or any chemotherapy, it's actually accelerating disease. And, I mean, would it be possible to mine, because obviously those drugs have incredibly variable effects on patients. And so even though in the need they might extend life, which they actually don't do that well in many cases. Right. Is there any way you could look and go back and mine data for, looking at the genotype and seeing whether the particular needs correlate with?
Yes. Well, I think that is something we want to do at some point. We also want to test some drugs, these drugs that can deactivate the mutant to make it wild type and see. Yeah, I forgot. Sorry. Okay, it's working. All right. So we can actually check that with the different drugs that we have. I mean, there was lots of progress in the p53 mutant p53 field, and the new drugs that came out, and it would be great to test it too, and at least in tissue culture if it doesn't work in.
You may want to talk to people about who are good at mining the data.
Yes.
Jim Costello, the science person, might point you in the right direction.
Yeah.
One of those groups.
Yeah, but as maybe you know or you don't know, it's pretty hard to have bioinformatic access, I think, over here.
You should talk to Tim Jones.
Yeah, well, he's my collaborator on one, but, it's just like you have one and you have 50 people. Yeah, it is. Thank you. Yes? Isabella.
Yeah, I was wondering if you could comment on what you mentioned about p53 having too much of it.
Mm-hmm.
It's also a problem.
So, are you asking about during development or more postnatally? Postnatally, there are other models where you can see some conditional models of, for example, taking off MDM4, and they will have, for example, heart defects. They will have cardiomyopathy and things like that much later in age. So they do have postnatal development, like in the nervous system, and they do have it too. So that's how having an extra level of p53, and it doesn't need to be too much, like twofold or threefold, just a little variable. But interestingly, I don't have the, and it's published in Cell, I think in 2014. If you have very little percentages of higher p53, it also boosts your survival, so it makes you live longer. At least that's in mice, you can see it, and it's beautiful because you can see how few percentages can just shift that survival curve to the left or to the right.
Will it be beneficial, like
I do, but they have, I don't think it can be a one thing to use alone. It's a combination therapy, and there are some phase 2 trials for like drugs of not mutant, but wild type p53. But you really need to know what's the status of your p53 and what type of mutation you carry, and having a personalized medicine approach to it. There is a different heterogeneity for, so I work also with the 172P model, which is also a hypomorph of p53 that interestingly maintain the cell cycle arrest function, but doesn't have the apoptotic function. And that mouse behaves totally differently, have different tumors, and like it lives actually very long, but still get tumors. It has different phenotypes than and different issues than these mice.
So there's no simple answer to p53.
No, it's only going to get more complicated. Yes, Bob.
The levels of MDM2 and MDM4 are
You mean in which model? You mean like when we take it off? It's constitutive. Yeah. Right. All right. Well, thank you.